I stand before you today with a message of hope. We do not need fetal body parts from aborted fetuses to achieve future scientific and medical advancements. Several alternatives exist that are abundant and more successful. After over 100 years of research, no therapies have been discovered or developed that require aborted fetal tissue. History has shown us that we never needed fetal tissue to begin with. In the case of vaccines, cells derived from aborted fetal tissue have been used in the development process, but fetal tissues have never been the exclusive means necessary for these breakthroughs. What does this mean? This means that if we stop harvesting fresh tissues from aborted fetuses today, it will not stop one person from being treated or vaccinated, nor will it inhibit the development of new vaccines or therapies going forward. Case in point, development of the original polio vaccine was first produced in monkey kidney cells. Most polio vaccines currently use the cell line Vera, or monkey, in the manufacturing process. Measles, mumps, and rubella vaccines can all be produced in non-fetal cell lines. And none of the 75 vaccines available in the U.S. are produced using fresh fetal tissue. Researchers have access to these ethical human tissue sources, which are available at several hospitals, academic institutions, and tissue banks across the nation with proper consent and IRB approval. For example, discarded cord blood from labor and delivery is stored in tissue repositories because it is a rich and plentiful source of stem cells for research, regenerative medicine, and transplantation. Stem cells from placenta, amniotic fluid, and even a mother's own bone marrow are even showing tremendous potential for in utero stem cell transplantation and treating the unborn before birth. In addition to stem cells, fully differentiated somatic cells from tissues such as skin can be reprogrammed to make induced pluripotent stem cells, or iPS cells, that possess an embryonic-like state. These pluripotent stem cells can then be further reprogrammed to generate organoids, also known as organs in a dish, which are 3D cellular structures replicating normal organ function in vitro for brain, liver, pancreas, intestines, stomach, lung, kidney, and eye. These organoids have proven successful in modeling complex mechanisms, including Zika virus and microencephaly, brain disorders, retinal development, microencephaly, brain disorders, retinal development, infectious disease, cancer, and many others. Post-mortem donations are another useful solution. One option is cadaveric tissue, from which stem cells can be isolated up to several days after death. And GCSF mobilized peripheral blood are all successful alternatives. In fact, human cord blood is perhaps the best scientific and non-controversial source for optimal human cell engraftment because it is abundant and more efficient and clinically relevant with respect to immune response. Other options include the ability of researchers to actually purchase humanized mice directly from commercial vendors such as Jackson Laboratories for immediate shipment. Such models include the human CD34 positive NSG humanized mouse developed using CD34 positive stem cells from cord blood, again without the use of human fetal tissue, as well as peripheral blood mononuclear cells for short-term experiments requiring mature T cells, including HIV infection. In conclusion, it is clearly evident that numerous alternatives exist and are available to researchers for studying some of the most complex diseases and disorders facing our nation today. I urge Congress and the Trump administration to stop using federal tax dollars to support aborted fetal tissue research and redirect funding to these alternatives.